students, my name is Neha Mittal. I am working as an assistant professor at Ramesh Institute of Vocational and Technical Education, Greater Noida. Today in this video lecture, I am going to continue anti-malarial drugs part 2. As in the previous lectures, we have studied about the etiology of malaria as well as their treatment strategy. So, as we know that malaria is a worldwide spreading disease and is affecting many people around the year. So, there are many categories of anti-malarial drugs in which today we are going to discuss about the quinolines, aminoquinolines, chloroquine, quinine sulphate and in the last Amodiaquine. First of all, we will understand about the quinoline. Quinoline is an aromatic heterocyclic structure which contains two rings. One is benzene ring which is of 6 carbon membered and another is pyridine ring which contains 5 carbon and 1 heterocyclic atom, that is nitrogen. So, 6 carbon atoms make the benzene ring, whereas 5 carbon atoms and a nitrogen atom make the pyridine ring. If we talk about the molecular formula, its molecular formula is C9H7N. Substitution of quinoline ring produces the few compounds which with the anti-malarial activity. As we see, if we do the substitution at the 4th position, it produces the 4 aminoquinolines, and if we do the substitution at the 8th position, it produces the 8 aminoquinolines. We will study one by one in the next slide. First of all, we will study about the aminoquinolines. Aminoquinolines are the amino substitution at the quinoline ring. As I told in the last slide that this is a quinoline, this is a quinoline ring, and if we do the substitution of amino group at 4th position, it is known as the 4 aminoquinolines. In the same way, if we see the substitution at 8th position with the amino group, it is known as the 8 aminoquinolines. So, these are the two categories of the aminoquinolines differing with their substitution of amino group. One, there is a substitution at 4th position, whereas in the second there is a substitution at 8th position. If we talk about the 4 aminoquinolines, these quinolines act against the zygentosides and gametosides form. The example under this categories are Chloroquine, Amodiaquine and Piperquine, whereas the examples under the 8 aminoquinolines are Pemaquine and Primaquine. So, we will discuss one by one. Now, we will start with the chloroquine. Chloroquine is a synthetic derivative of quinine. It was first synthesized by the German scientist Chloroquine in 1934. Chloroquine is a 4 aminoquinoline with anti-malarial and anti-inflammatory action. If we see the substitution, we will find that it is substituted at position 4. It is substituted at position 4 by the compound. If we see the numbering 1, 2, 3 and 4. So, at position 4, it is substituted by the diethyl amino. There are the 2 ethyl compounds 1 and 2. So, diethyl amino 1 methyl butyl. So, here we see that there is a presence of methyl group at position 1, then 1, 2, 3 and 4. So, here we can say that it is a substituted at position 4 by a 4 diethyl 1-methyl butyl, and there is a substitution at position 7 by the chlorine molecule. If we talk about the mechanism of action, so hemoglobin is broken down into two components, that is heme and globin. Globin part is utilized by the malaria for the nutritional purpose, and heme part is the toxic to the malaria. So, there is an enzyme named heme polymerase which converts toxic part of heme to the non-toxic part, that is hemojoin. So, here our target is heme polymerase, which is responsible for converting the toxic material to the non-toxic material with respective to plasmodium. So, here are the drugs chloroquine, quinine and mefloquine. They inhibit the function of heme polymerase. So, there is a accumulation of heme inside the malarial parasite, which is very toxic to the malaria and simultaneously causes death of malarial parasites. Here we will talk about the method of synthesis. If we see the synthesis of chloroquine, it is made up of 3 steps. First, there is a synthesis of quinoline. First of all, this quinoline ring will be synthesized. The second, synthesis of this side chain, and last, condensation of both quinoline ring and the side chain. So, we will discuss one by one. First of all, we will understand the synthesis of quinoline ring. For the synthesis of quinoline ring, we will take the meta-chloroaniline. Here there is a chlorine at meta position. It will react with the ethyl oxaloacetate, and with the liberation of water molecule, it will produce the intermediate compound. There is a liberation of H2O. So, this NH2 will liberate H, and this will go and bind with this nitrogen. So, by this way, this is the formation of this intermediate. Now, in the next step, at high temperature around 250 degrees Celsius, there is a process cyclization takes place. In the cyclization process, there is a formation of ring with the removal of C2H5OH. Furthermore, we see that in the presence of caustic soda, water and HCl, there is again removal of another molecule of ethyl alcohol, forming the 7-chloro-4-hydroxy quinoline 2-carboxylic acid. Here we will see that there is a chlorine group at 7th position, hydroxy group at 4th position, and the carboxylic group present at the 2nd position. Next, there is a liberation of carbon dioxide. So, there is a formation of 7-chloro-4-hydroxy quinoline. Now, there is no carboxylic group present, as it has removed in the form of carbon dioxide. In the last step, substitution reaction takes place in the presence of POCl3, and there is a removal of OH group and addition of chlorine Cl. So, this is a 4-7 as 1, 2, 3, 4. This is the 4th position, 5, 6 and 7. So, there is a formation of 4,7-dichloroquinoline. So, this is the first step where we form the 4,7-dichloroquinoline. Now, we will see the synthesis of side chain, which is a second part of the chloroquine synthesis. So, here we will take the compound 5-chloro-2-pentanone and will react with the diethylamine. So, there is a liberation of HCl molecule, this H and this Cl will get out from the reaction, and there is a formation of diethylamine with this compound. This is an intermediate compound which will formed by the reaction of 5-chloro-2-pentanone and diethylamine. Now, next we will see the reduction reaction. So, in which there is a formation of COH, means this O double bond O will get reduced to the OH group. Next we will see that there is a formation of 2-hydroxy-3-diethylamino-pentane. In the last step, we will see that there the substitution reaction takes place. In the substitution reaction takes place, we take the two compounds, one is SOBr3 and second is NH3, that is ammonia. So, there is a formation of 4-amino-1-diethylpentane with the liberation of HBr. So, here is a synthesis of side chain. Now, the third step is condensation of both 4,7-dichloroquinoline and the 4-diethylamino-1-methylbutylamine. Here we will see that if we do the condensation of these two at 180 degree Celsius, so there is a formation of chloroquinoline. So, by this we can understand the synthesis of chloroquinoline which contains the three steps. In one step we make the synthesis of 4,7-dichloroquinoline. In the next step we see the synthesis of side chain, and then in the third step we do the condensation of these both two compounds. Then we will see the structure activity relationship of the chloroquine, in which we will see the nitrogen of the amino group attached with the chloroquine group, means this nitrogen group is responsible for the basic character of the drug. Next we will see the tertiary amine in the terminal. This tertiary amine is very much important for the drug activity. Third, we will see the spacer length is very much important for their antibacterial activity. If we shorter the length as well as if we longer the length of this spacer, it affects the antimalarial activity. Next, we will see that presence of small electron withdrawing group at 7th position is very much important for the drug, as it is responsible for the inhibition of hemojoin formation. Next, we will see that if at 3rd position there is an addition of methyl group, it leads to the decrease in the antimalarial activity of chloroquine. Next, we will study about the uses. So, here we see that it is effective against the asexual erythrocytic form, but it is not active for the hepatic form of the parasites. Next, we will see it is an important drug for the treatment of malaria, but it is not recommended for the prophylaxis action, means it is not used for the prevention purpose. Chloroquine is used to treat rheumatoid arthritis, different parasitic infection and hepatic amoebiasis. This drug is safe if it is used in an appropriate dose. If we see the side effects, then we will see the gastrointestinal and visual disturbance, देखने में problem होना, visuality में आपकी problem आना are the common side effects which are seen with the chloroquine. Chloroquine phosphate is available in the tablet form, and it is also used for the treatment of coronavirus infection. However, it is based on risk and benefit ratio. Next, we will see about the side effects. So, nausea, vomiting, diarrhea, headache and abdominal cramps were the general side effects which were reported with the chloroquine. Next drug is Amodiaquine. Amodiaquine was first made in 1948. It is a 4-aminoquinoline congener of chloroquine used in some countries for the treatment of malaria. It is somewhat similar to 4-aminoquinoline, but there is a difference that it is having a para-hydroxy aniline aromatic ring on its side chain. Means this extra ring is incorporated in the structure of chloroquine, which is named as Amodiaquine. Amodiaquine is effective against some chloroquine resistant strain, particularly Plasmodium falciparum. It is not used as prophylaxis due to the development of resistance, agranulocytosis or severe liver dysfunction. If we talk about the metabolism in the body, so Amodiaquine is converted in the body to the compound des-ethyl-aminodiquine. What happens? There is a, if we talk about the Amodiaquine, so there is a presence of 2 ethyl group, but when it gets into the body, it metabolizes to the another compound, that is des-ethyl-amodiaquine, in which 1 ethyl group is removed and the hydrogen atom is incorporated. If we see that the mostly side effects are likely due to the presence of the metabolite des-ethyl-amodiaquine. Amodiaquine is an orally active 4-aminoquinoline derivative with antimalarial and anti-inflammatory property. Mechanism of action: Amodiaquine is able to inhibit heme polymerase activity in the body. This results in the formation of free heme, which is toxic for the parasite. Amodiaquine is converted to the active metabolite des-ethyl-amodiaquine, which is responsible for the production of heme, which gets accumulated inside the plasmodium and ultimately result in the death of the plasmodium. Next compound we will study quinine sulphate. Quinine sulphate is a bitter taste anti-malarial drug among the alkaloids of cinchona. In cinchona bark, anti-malarial activity are due to the quinine, quinidine, cinchonine and cinchonidine. Quinine is the sulphate salt form of the quinidine alkaloid isolated from the quinine. An alkaloid derived, this is derived from the bark of cinchona tree. Quinine sulphate is obtained after many recrystallization process in the lab. If we talk about the properties, so the taste is very bitter. It is white crystalline powder, odorless, fine crystals. If it gets exposure in light, it becomes darker in color. Solubility is in alcohol and very less soluble in water. If we talk about the decomposition, when heated, then it emits very toxic fumes of nitrogen oxide and sulfur oxide. If we talk about the dose, then the standard dose for the treatment of uncomplicated falciparum malaria is 648 mg orally every 8 hours for 7 days. For pregnant women, the normal dose of quinine sulphate for 7 days is recommended. The pediatric dose, if we talk about the kids, then it is 10 mg of quinine sulphate per kg orally 3 times a day for 3 to 7 days. Uses for the treatment of uncomplicated malaria in adult, बड़े लोगों में the dose of Qualaquin is 648 mg, but in the kids and in the pregnant women the dose differ. There is a medicine Qualaquin should be taken with food to minimize the gastric upset. Sometimes we see there is a symptom, there is a side effect of gastric upset. So, we can take this drug to minimize the gastric upset side effect. Side effects are fever, ठंड लगना, पसीना आना, flushing, चक्कर आना, asthenia and hypersensitivity reactions. These are the few side effects which were reported with this drug. Thank you so much. I hope you learned a lot from this video lecture. Thank you so much.